So the goal of this whole project is to use RNA interference to knock down the gene, or to knock down the enzyme hyaluronidase in bee venom, due to the fact that it could cause a varying severity of allergic reactions in people who are allergic to hyaluronidase. So many of those who are allergic can have very lethal reactions to it. So the whole point of this is to prevent that from happening. But by knocking down the enzyme in the gene, we can prevent these anaphylactic shocks from occurring while also still allowing the bee to have their defense mechanism so that they can defend for themselves and their hives. Many of those who are allergic to hyaluronidase have allergic reactions or anaphylactic shocks when exposed to this venom. Their immune system is basically overreacting to the foreign antigen, causing them to have terrible symptoms like swelling or not being able to breathe. So the whole point of this is to silence the enzyme hyaluronidase, the whole point of this project is to silence the gene which produces the enzyme hyaluronidase so that they can still defend themselves with their stinger, but prevent people from going into anaphylactic shock.
Okay, so first we would identify the gene sequence that we want to silence. This will be our gene sequence right here. Bee venom contains several biologically active peptides and 2 enzymes that include hyaluronidase. The gene sequence below is the gene found in the bee venom, hyaluronidase, that we are trying to silence. Next, we want an RNA extraction. To do that, we have to dissect the venom glands, then take the various bee tissues, including the venom glands, and blend into a buffer solution containing 4.2 mole guanidine isothiocyanate, 100 millimole sodium acetate, 2 millimole EDTA, 2% percaptoethanol, and 2% 1-butanol. After extracting with phenyl chloroform isomal alcohol, the water phase will be acidified with 2 molar sodium acetate, extracted with phenyl chloroform and precipitated with isopropyl alcohol. The pellet is then dissolved in 6 molar urea, 100 millimole heps, pH 7.2, and precipitated by adding 0.5 volumes of N molar lithium chloride. Lastly, poly-A-rich RNA is isolated by chromatography on
cellulose.
Yeah, cellulose. So our target gene is extracted from the venom glands, dissected from bees. mRNA is extracted from the venom gland tissue. The resulting solution of mRNA is purified with a solid-state hybridization with a commercially obtained probe.
Okay, so at this point what we have is just a solution of pure mRNA. To get that, we have to turn that into DNA first, and to do that we do an RT-PCR using only primers for our particular gene. That'll weed out the unwanted genes that would also have been transcribed through the RNA. With that, we then do a restriction digestion in order to break those into individual parts, since for RNAi, you don't want the full gene, you just want small parts of it, and some of them will have better affinity than others for what we want. So we use DNase 1 to digest it into reproducible fragments. Those fragments will then be separated using gel electrophoresis in order to isolate the individual fragments. Those will then be ligated with an siRNA adapter sequence, so that instead of just coding for the gene, they will instead code for a gene that gives our target plus a hairpin so that it forms double-stranded RNA. And then that gene will be spliced into a baculovirus sequence. Baculoviruses are insect viruses. They were actually originally found in bees, and they've been used as a recombinant tool for many different insect species. They came from bees, so we know they'll work for bees. That's why we picked it. And then what we'll do there is then we'll begin screening the individual sequences to see which one actually works better. This part will be done by a firm that we'd hire to do this. But for an idea of what they'll actually do, for screening, they take the individual viruses. They, of course, replicate them using typical bacterial cells. And then using those, you will then infect insect cells that are modified to have... Okay, at this point we have a solution of pure mRNA from the tissues. We will run an RT-PCR using primers for our target gene so that we can get a whole bunch of copies of our gene in DNA form. We will then run a restriction digest on it using DNase 1, which will break that all the copies we have into specific size pieces. Those pieces will then be ligated into to have a siRNA adapter sequence, so that the code for the double-stranded RNA hairpins instead of just the gene. That RNA library will then be ligated into empty baculovirus sequences. This will be done by a company, not us. And then those will have to be screened to determine which one is the most effective, since for RNAi, different parts of the sequence are more effective at binding than others. The way that screening will be done is using an insect cell line that expresses our target gene, hyaluronidase, will be dosed with the virus, different viruses since we have them all separated. Whichever ones are more effective at blocking hyaluronidase production, that's the one we will use for the final product. We will then mass produce that specific virus that was extremely effective, and then that will be mixed with water to try it in actual live bees in the lab, in sugar water. The bees will be fed the sugar water, and an RT-PCR will be performed on extracts from those bees to determine if gene function has been silenced for the hyaluronidase gene. If it is, then we can start field trials by putting it into sugar water again and putting that near beehives, such as in apiaries and so on, with the ultimate goal being no harmful effects on the bees, and the bees are no longer able to kill.
All right, so for the ethics of this project, an issue that is we're targeting the bee venom, but the only defense mechanism that bees have is their stingers.
The vectors, so their venom won't be able to sting humans. Well, they'll still be able to sting us, but we won't go into anaphylactic shock to where it's lethal for us, where we would die from their venom. So let's just say that once we did inject them, if something went wrong, because mutations can always happen, nothing worked perfectly. It's all about trial and error. So if we did inject them, purposely trying to make their venom not attack humans, but let's just say something went wrong and they stopped making their venom overall, then that would be bad because the male drones won't be able to protect their hive.
So an issue with using virus vectors is that these viruses, you can't really trust them. They can end up going rogue, attacking the bees, changing their physiologies, or just end up killing them, and just like viruses, they can spread and take down entire populations, which can have be really bad considering that bees already have an issue with their populations. Another thing too is that there could be some unintentional side effects of these vectors. Like for example, Africanized bees. Initially, scientists were just trying to create a bee that produced more honey. What ended up happening is they actually created a more aggressive bee that ended up breaking out, escaping, and invading the whole Americas. So a situation similar like that could be an issue if we're working with a vector. So overall, the chances of success with this project are still not totally sure. It's never been done before, and I don't believe any experience similar to this have been done before. But we should always try, as they always do in science. That's how you make progress.